right, good morning, everyone. Okay. So, I guess my getting little announcement things out of the way at the beginning here. For any of you who have been using the lecture recordings that are on Moodle, you might be happy to note I picked up a new mic last night. This is not it. This is still the old setup. I have not tested the other one sufficiently to be sure it's not going to sound worse. But it should help with some of the minor issues we've been having. This one has buttons on the back that I didn't realize. So I had bumped one of them. So one of the recordings is a little bit less clear than I would have liked. But hopefully with the new one, that shouldn't be an issue. And it's going to be portable, so it'll be right here, so less background noise and all of that. So we have that to look forward to next week. But for today, we're going to start talking about the digestive process, or I guess continue talking about it, because we had previously been talking about things like our preference for taste and the fact that we have a little bit of a biological predisposal to liking certain tastes for certain reasons. But now we're going to look at the actual process of starting from eating something and then getting nutrients from that. So it's interesting to note that the fact that we eat sort of large meals all at once and then go for very long periods with no food in between is actually pretty special. Some animals will eat sort of all the time. They will constantly graze so that they don't have to worry about storing and retrieving nutrients. But we and a bunch of other species have a mechanism in place where we can eat really large meals, store up some of that nutrients for later, and then have time in between where we don't have to eat, but we still have all the energy that we need to function normally. So we have these intermittent periods of fasting in between larger, more calorie-dense meals, as opposed to other species who might sort of gradually graze all day on less calorie-intensive meals, but they don't have those fasting periods in between. All right, but for us doing that, having that ability to eat all at once and then not in between means that we have to have a pretty specialized system to permit us to do that. Because we've talked about the fact that our body uses up a lot of energy, and especially our brain. Our brain uses up a ton of our resting energy. And so if we're not actively eating, it's still burning through a lot of resources. So we need to be able to keep some kind of regular access available to it. But we will get to that system. We're going to start off with the start of our digestive process. So this is when you're eating. We've already mentioned that saliva is there to basically dissolve our food. We talked about it in terms of dissolving things that we can then pick up as tastes on our taste buds, but it also dissolves the food so that it can start the digestive process. Our saliva even has enzymes in it that help us start breaking down things, especially starchy things. I don't know if you ever did this in elementary school, but I know my classes did, where they would give you an unsalted cracker and they'd get you to put it in your mouth. And at first it just tastes like a gross cracker, but eventually it starts to taste a little bit sweet. And that's because your carbohydrates in the cracker were being broken down into glucose or sugar molecules. So you can actually test that yourself if you have some unsalted crackers at home. You can still figure it out with salted crackers. It's just harder to tell the sweet and the salty apart. But I always remember that little trick. So it's sort of a utility thing where it gets our food sort of lubricated and capable of being swallowed and passed into our digestive tract, but it also starts the process of breaking stuff down. And then our teeth make everything into small enough pieces to be dissolved and broken down. So it's all a pretty good start there. And then in our stomachs, we have all of our stomach acids, so mostly hydrochloric acid, but there's also a mix of pepsin. So that's another chemical that actually reacts. It's only really functional in higher levels of acid, so it's specialized to work with stomach acid, which is perfect. And that helps us break stuff down again. And then from our stomach, we have a gradual release into our intestine. Actually, I can be on this page to show you. So if we started at the top with the mouth, down into the stomach, And then the stomach is able to be a little bit of a reservoir. We don't dump all of the food out of our stomach all at once. It's going to be a more gradual process. And the reason why it's a gradual process is because it allows digestion to occur at sort of a steady pace instead of all at once. And so most of the digestion that we care about is going to start occurring right in here. It's really hard to see what I'm pointing at over there. So it's just the first couple of centimeters of our small intestines, or the duodenum, or duodenum, depending on how you put your emphasis. I've heard it both ways, and I don't actually know which is right. But that section, it's about the first 25 centimeters or so of your small intestines. That's the first part where we start sort of pulling that nutrients out and start bringing it into our body for use. And so in there we already talked about the carbohydrates having started to be broken down in our mouth, but that process continues. So carbohydrates get broken down into simple sugars. So just based on what carbohydrates are made up of, as you separate those longer chains down, you get sort of much smaller sort of individual links of those chains, and that tends to be glucose. Sometimes you'll have sort of 2 or some of the smaller chains that are fine, but we care mostly about glucose because that's going to be one of our primary sources of energy, especially for our central nervous system. So that's what our carbs do. Proteins get converted back into amino acids, which kind of makes sense if we think about the fact that our bodies will build up chains of amino acids when we're trying to make proteins in our cells. Makes sense that if you wanted to then use those nutrients, you could break them down into those constituent components. So we have lots and lots of amino acids, and we'll pull those back out of proteins. Any fats that we eat get transformed into fatty acids and glycerol, so 2 different chemicals. And anything to do with fat usually involves the liver. So digesting, storing, telling the body about fat is usually going through the liver. And so all of those different little pieces of what we've eaten, all of those constituent parts get absorbed through the wall of the intestine, or they'll get and then transported to the liver. So they'll go through the wall of the intestine along the hepatic vein, which is just the vein that connects to the liver, and then the liver is going to figure out what it's doing with stuff. And so at that point, we're still in a very liquid state. So between our saliva and all of the acid in our stomach, we have lots and lots of fluid that's been incorporated with all of those products. And it needs to be. In order to keep flowing down our intestines, there needs to be quite a bit of water. But as we talked about last time with regulating thirst, our body it needs to do a good job of conserving water because we need a lot of water in our bodies, so we can't afford to lose those large amounts of water. And so that's why the rest of our intestines, basically all of our large intestines, are completely set up in order to reabsorb water from all of the leftover stuff. So we end up trying to pull back as much as we can in order to sort of not lose that much, not to be dehydrated and not have to go out and find more water to sort of replace it. So that's the very basics of it. So I did talk about the duodenum or duodenum as that initial 25 centimeters of our small intestine. That's where most of the digestion's occurring. Cool. We also have an interesting portion, which is the area postrema. And this is a part of our brain that is, again, outside of that blood-brain barrier. So we've mentioned the fact that our blood-brain barrier keeps toxins and bad things out of our brain. But if we wanted to monitor things that might be in our blood, then you can't have the brain necessarily in direct contact. So you have to find a way around that. And so there are certain points that will be outside of the blood-brain barrier in order to monitor things. So we keep more of our critical core functions internally, and then there are sort of bits that project to allow us to monitor. And this area postrema is specifically there to allow us to identify any toxins that have slipped past all of our other defenses. So if we're eating something, we have a bunch of different ways that our body can tell us that it's bad for us. So the first is taste. If things taste really awful, especially rotten, then we're not going to want to eat it, and it might cause us to spit it out right away. That is something that we can learn. There's also a little bit of innateness there, where certain tastes just don't sit well with us. If we can get past that, if there's still something bad in it that passed that taste test, then our stomach has a process that'll check for toxins as well. And if your stomach is irritated, specifically the lining of your stomach, is irritated by what you've eaten, you will actually throw up. So if you've ever had food, maybe it was something that had gone a little bit off, maybe you were eating a new kind of food and it didn't sit well with you, if your stomach sort of did the reaction and you threw up shortly after, that would be that portion of your body getting to that. So your stomach said, nope, this is not sitting well, it's probably bad, just to be safe, we're getting rid of it. But sometimes we have toxins that don't irritate the stomach lining. So we still have this last line of defense, and that's the area postrema, where if something has gotten through our taste test and it's gotten through our stomach, in the first step of that digestion, parts of it start getting absorbed into the blood. And that's why the area postrema touches our blood. It passes that blood-brain barrier to monitor for toxins in our bloodstream. And if it notices that there are bad things in our blood, it has a very strong and sometimes violent reaction, which is to induce vomiting. And it will occasionally induce projectile vomiting, especially if it's something that it deems very threatening to the body. And so this happens fairly quickly. So as soon as it detects it in the blood, again, better safe than sorry. And so you can end up throwing up too much very, very quickly just because of what it's detected in the blood. Mm-hmm.
So there like a certain foods or like edible stuff that is not very good to taste, but it has like for instance the thing in my mind was neem. So it has a bitter, very bitter taste, but it has a lot of antioxidant properties. Is there some way you would be? How would you explain it? It's being bitter, but still antioxidant. Like, is there a connection to it? Is there some relation you could?
So just being bitter isn't necessarily a bad thing. That's why, like, there are like rotten flavors are one of those things that will automatically set us off. But other things, like being too bitter, we might have an initial sort of, I don't know that I like that, but we can learn to like it, especially if we then eat it and don't get sick. So the taste part is one of the easiest to overcome because we can kind of consciously override it. That's one of the problems with children, right? They don't want to eat anything that tastes funny, so they don't like broccoli or Brussels sprouts or anything with a strong flavor. But if you can convince them to eat it and they see it doesn't make them sick, they can eventually like it, and then that taste doesn't bother them anymore. So you can kind of trick your brain into overcoming some of your defaults because they're not hardwired, they're just sort of suggestions, if that helps. Cool. All right, so those are some of our defenses. That's the first stages and the basic overview of sort of pulling nutrients out of food. Next, we can talk about some of the stuff that's been pulled out, and we've kind of dropped some of these names already. So we mentioned glucose as being something that is broken down from our carbohydrates. So it is a simple sugar. It's going to be a common metabolic product from carbohydrate digestion. You'll sometimes get things like dextrose and other sugars, but glucose is our primary element that we want. Amino acids are from proteins. We said that made sense. Fatty acids are what we convert fats into, and that's more of an biologically acceptable or biologically available version of the fats. And then glycerol is what happens when fats are digested not in the intestine but in the liver. So fatty acids and glycerol are both sort of our byproducts of digesting fats, just depending on where they were processed. And we tend to break down our fats in all of those places, wherever we can get them. Okay. So that's the setup of it. Next, we can talk about that absorptive phase and the fasting phase, so the part where we're actively eating and then the part where we are not eating, and not just on what's happening with the food itself, but what's happening in the rest of our body. So we covered the fact that stuff's being broken down, mainly in our small intestines. A little bit of it gets sent to the liver to be processed there. That's fine. But with this absorptive phase, while we're actually absorbing food or nutrients from food, what else is going on in the body? And so here we're going to see that we have a neat sort of trade-off, where we had talked before about our sympathetic and parasympathetic nervous system. So our sympathetic was, it's when we're trying to sort of burn excess things. We're trying to take stored nutrients and turn them into something that we can do with. So things like fight or flight, where we need to be in action and we're pulling energy to stay in action. But our parasympathetic, I've recently seen it referred to as, instead of fight or flight, it's the feed and breed side of things, which I thought was weird, but it kind of makes sense in terms of what it's doing, where while you're eating, you're storing stuff away for later. You are sort of being stable and stationary and saving those resources for use at a later time. You're not under threat, so you don't have to burn them immediately. You can put them aside and be prepared for a later occurrence. So it kind of makes sense with the name there. And so I'm going to hop a little bit between this diagram that sort of shows the simplified version of both of these phases, and then the text that describes some of these phases, and then we'll kind of hop back and forth a little bit. But if we wanted to give a technical name, this absorption phase is the few hours after a meal while our body is still actively processing our food and we're living off of the nutrients that have arrived into the digestive system. So this is when you're surviving, your body is functioning using that newly brought in resources, all of those sugars and fats and all of that, those amino acids. So we don't have to pull from our stores because we've eaten something and now we can live off that for a little while. And so during this process, we have insulin as our really important hormone. And insulin enables the cells in our body to take up glucose, so to pull glucose from the bloodstream and burn it as an energy source. So this is really neat because insulin, it isn't necessarily doing anything to the glucose itself, it's just making it so that the glucose can pass into our cells. And then when our insulin levels drop, after we've sort of stopped eating and there's less of that free-floating glucose, if there's no insulin in our system, our body's cells won't continue pulling glucose, even if the levels start rising, which will become important a little bit later when we're pulling from our stores. So insulin is what lets us have glucose move into our cells or not. Okay. So we did the glucose is increasing because we're digesting things that are releasing glucose. This leads us to that parasympathetic system turning on. Our parasympathetic nervous system will activate our pancreas, which is how we get that insulin secreted into our bloodstream. And so insulin allows the glucose to enter our body cells. And this is specifying body cells because we're going to have a distinction between the cells in our central nervous system and then the rest of the body later. So if it seems weird to talk about cells in our body as body cells, it's just because it means non-central nervous system cells. It's just the text isn't super clear about that. And so we talked about the fact that we can have some glucose starts to get stored in the liver, and our muscles as well. So we can turn it into glycogen, which is going to sort of be a longer, stabler form of it. So while there's excess, we're going to store it away. And we can store some of our fats. Again, if we have access, we can store it as triglycerides in our adipose cells. So adipose cells are just fat cells, more of that sort of more chemically stable, long-term storage format. So that's our absorptive phase. And we can mention that with diabetes, because if you ever mention insulin, it should pretty much immediately make you think of diabetes. This is when we have issues with insulin in one way or another. So diabetes is a condition that happens when either type 1, the pancreas isn't able to produce enough insulin, So you don't have enough insulin present in your blood in order to let your cells take up that glucose that's free floating. Or for type 2, if your body's tissues are relatively unresponsive to insulin. So this could be a problem with insulin receptors. So you produce enough insulin, but your cells don't respond to it the way that they should. So we can have an issue with either the presence of insulin or with how we react to it. That's the difference between type 1 and type 2 diabetes. And then we talked about glycogen as a converted form of glucose, and that's more of our stable type of glucose that we can store for use later on. That's going to be more of a short-term reserve. Things like fats are going to be a longer-term reserve, but again, we'll see some of that coming up. Okay, so that is our absorption phase. Now we're going to look at our fasting phase, and so this happens as that glucose level in our blood starts dropping. And it makes sense. If we are using it to run our cells and we're putting some aside for storage, we're going to burn through it fairly quickly, especially once we've stopped eating and we're doing other things. So that's why the fasting phase is called that, because you're no longer eating, and we can go for pretty long periods without eating. And again, I'm probably going to hop back and forth between those two slides a little bit, but that's all right. And so this fasting phase, it's the period where our blood glucose drops and our body has to go back to relying on existing energy stores. So there's no longer enough new incoming nutrients from what we have eaten, and so we have to go back to relying on what we had stored away while we were in that absorptive phase, while we were putting stuff aside for later. So we're going to start pulling things out of those stores. So as those glucose levels drop, our insulin levels are going to be dropping as well. And then instead, we're going to have a new hormone, and this is glucagon. And it's a hormone that causes our liver and skeletal muscles to start transforming that stored glycogen, first one was right, back into glucose. So that's why we had talked about glycogen, pronunciation today, glycogen as sort of a shorter term storage one, because that's our preferred method of pulling out glucose again. So we store it away while we're eating, and then as soon as we're done eating, that's our preferred source to go back to. So it's pretty easy to convert back into glucose, and we can bring that back into our bodies. And then if I come back here, so as that glucose is dropping, so we said before, the increase of glucose was activating the parasympathetic system. The decrease of glucose switches us over to the sympathetic system. So that's where we're burning things actively. And so again, our pancreas is involved, but instead of secreting insulin, it's now going to secrete glucagon, and it's going to transfer our glycogen into glucose. And this is where it's important to note that because our insulin levels are back down again, we're not producing lots of insulin anymore, that means that the cells in the rest of our body are no longer able to pull that glucose out of our bloodstream. So even though this glucagon has released new from the store as glucose into the bloodstream, the rest of our body isn't able to take it up yet. So instead, it's only going to go into the cells that are sort of being prioritized. So our central nervous system, specifically mostly our brain. Like I said, most of our energy, especially at rest, gets diverted into the brain. And so for the rest of the body, that's going to start releasing some of our stored fats. So we would have fatty acids are going to be released, and that would be for the rest of the body to work on. So they don't get the easily accessible glucose. That's reserved for the brain that needs that fast and consistent flow. But the rest of the body will burn sort of fatty acids. And then we can also have glycerol. So if we're sort of getting rid of our fat stores, if we're burning what we had stored temporarily, we're going to send some of our glycerol converted back into glucose up to the brain as well. So there's a couple of different processes. And it makes sense. If we didn't know when our next meal was coming, or if we weren't certain that we were going to have a next meal soon, we'd like to have lots of different types of stores that we can burn. So The glycogen is a good one because it's quick and easy. There are harder ones to get to. We can use the glycerol, but if we're really desperate, we can start doing things like breaking down some of our muscles in order to use sort of the stored proteins and stuff there and turn stuff back into energy that way. So we have lots of different backup systems just in case. But in a daily cycle where we eat fairly often, the recommended 3 meals a day, we would cycle back and forth between the absorptive and the fasting phases pretty quickly and not have to resort to anything too drastic. Okay, so that was that guy. Okay, so that's the process of storing and retrieving different things that we can burn for energy, and that was fairly straightforward, at least hopefully it was fairly straightforward. But of course it gets a little bit more complicated than that, because there are other things going on in the background where we don't necessarily just eat because we're supposed to eat. We also eat because we get signals from the body that tell us that we should be eating. So we have a whole system set up that helps us regulate our hunger, which should encourage us to eat whenever our levels get low, and sort of time things correctly so that we eat at the right time and in the right amount in order to maintain this system. So we're going to talk about a couple of areas of the brain and also a couple of different chemicals that help our body regulate these things. And so we'll talk about first sort of activating our hunger or turning on our desire to eat. And then we'll look at the end of how do we stop eating. So what are the signals involved in both of those processes? And so first we have our arcuate nucleus, which I, for the longest time, read as accurate. And then I couldn't understand why the acronym was ARC, but it's arcuate. So hopefully I don't say it wrong. But this is sort of our command center. This is the master of our hypothalamus, and it's going to control and integrate information about food intake. So that's going to be really important. And we know that it has a strong role in this food intake control because it has a lot of different kinds of neurons that connect to the parts of the brain that are involved in regulating food and hunger and things like that. So first we have our NPY/AGRP neurons, which is absolutely a mouthful. So NPY are just neuropeptide Y, and then the AGRP is a GUDI-related protein. So those are neurons related to our neuropeptide Y and that GUDI-related protein. You are fine just knowing the acronyms because that is quite a lot of word to remember. So these are neurons that are going to increase eating behavior and help reduce our metabolism. So it's specifically going to release some neuromodulators, so chemicals, things like our neuropeptide Y and that agouti-related protein. So this is what's helping us switch from our fasting system back over to the absorption phase. So we're going to release these neuromodulators, things that will modulate what's going on in the body, and it's going to tell our body to start using what's already there instead of burning what's stored in our resources or in our sort of reserves. So this is saying stop metabolizing what's already there and focus on what's coming in, the new stuff that's coming in. And so this switchover is part of what increases or encourages us to start eating. So that's the start of our hunger signals. Okay, so then we have our POMC neurons. And again, we have another lovely word. So this is pro-opiomelanocortin. So definitely stick with the POMC for that one. But it's just neurons that release that POMC, and it ends up activating the satiety neurons and decreases eating. So The NPY and the other ones at the beginning, those start those signals. They tell you to go find food, that you're hungry, that you should start eating. And then the POMC neurons say, okay, we've eaten now. We have enough in our system. We can go back to metabolizing our reserves. So that kind of gives us the idea that, okay, this arcuate nucleus has both of these types of neurons connecting to it. So it must be involved in both turning on and turning off hunger. Mm-hmm.
I heard a while ago that neurodivergent, but specifically like people with autism and ADHD, have issues with feeling hungry, like introspection in general, like being able to tell what their body needs. And I'm wondering if you know what that relates to in the neurons and in the brain.
I don't know specifically, because I know it's not just food specific. It's also, oh, you know?
I don't know exactly like what, but I know that it relates to attention and boredom. They have issues sensing their hunger, but it relates to when they're bored, they feel, they don't start to feel the hunger, but they feel that impulse to eat.
Interesting. Because I know there's also some talk of things like it not occurring to you to do the actions that will solve a problem. So things like if you have a headache, it doesn't occur to you to take Advil to help with it. You know that Advil would help, but you can't make that connection. So it might be that part of connecting a feeling of hunger to actually that being hunger. So it might be a misinterpretation, or even the fact that you don't assign it sort of enough importance to pay attention to it. But I only found out about that recently, so I don't know too much on it. But that's kind of what my thought would be. Be interesting to look into it some more. All right. Okay, so next we're going to talk about our lateral hypothalamus. And so this is going to be the part of our hypothalamus that's just sort of lateral to, I guess, the back, I guess, is where it is. I don't really care about the locations. We're just going with, it's the lateral hypothalamus. So this initiates eating and helps control aspects of our feeding behavior and metabolic process. So this seems to be, kind of makes sense. So if the arcuate nucleus is in the hypothalamus, this is sort of the larger area around that that's also going to be involved in the rest of our regulation. So if we were focusing in on that arcuate nucleus as a smaller area that does specifically sort of turning on food-seeking behavior and then telling us that we're satiated at the end, this is a broader area that's going to be controlling sort of the eating itself and then some of our metabolic responses to eating. So this is specifically involved in things like chewing and swallowing. This is why we salivate when we're eating. So you know that you automatically start salivating when you eat. Sometimes you even start salivating when you think about eating. I saw one of the best tricks for that is if you have a dry mouth, the best thing to think about is eating like crunchy dill pickles, and it will immediately make you start salivating. And for whatever reason, it seems to work. It's also involved in our secretion of our gastric juices, and then it triggers that insulin production and all of that. So this is going to help us sort of start thinking about eating. It's going to make us move around a little bit more to get food, but then it also regulates all of the actions involved in consuming that food. So whereas our arcuate nucleus neurons, those ones were more on the you're hungry, you should start seeking food side of things. This gets us more into the, okay, seek food and consume it side of things. And this part of the hypothalamus is also involved in some of the weird interactions that we have with eating and reward. So it connects to the reward center of our brain, and that's why sometimes when you eat, it makes you feel better, because certain foods will activate that reward center more or less. Okay. And then we have orexin, and this isn't an area of the brain. This is now specifically a neuropeptide. So it's going to be one of the chemicals that our body produces, and it increases eating and increases wakefulness whenever we're food deprived. So when we haven't eaten for a while, it's going to make us want to eat, and it's also going to make us less tired. So if you've ever noticed that you're getting ready for bed, you're exhausted, but then you realize that you're hungry, and all of a sudden you can't think about going to sleep, you have to deal with being hungry, that's orexin. It's making you sort of too focused on something else to be able to sleep. You're going to be awake until you deal with that more pressing problem. And evolutionarily, it makes sense because you wouldn't necessarily want to go to sleep while you're very, very hungry, because there's no guarantee that you would find food immediately in the morning, especially after a long period of fasting. So if we weren't in a society where you have a kitchen with food in it, that makes sense. And so it does this because it's specifically exciting those NPY neurons, so the ones at the very beginning. It excites those NPYs, and it inhibits the POMCs. So it's specifically biasing that arcuate nucleus towards the fine food side of things. So orexin is going to sort of push our system in that direction. It's going to push us to go out and get food, and it's going to turn off the signals that you're not hungry. Okay, and then because of this role, if you have something that is an appetite stimulant, so something that would increase your appetite, you might actually see it called orexigenic. And it's because they usually involve orexin, because that's that trigger that you should be stimulating your appetite. You should be eating. You should be finding food. Okay, so that's the first slide. Actually, do I have in this image? Yes. Okay, perfect. I do have a visual. Again, I don't test on the locations, hence not caring too much where stuff is located. But if you're a visual person, this diagram is a great way to see where all of those acronymed locations are located and what they make connections with. So this would be a good thing to have nearby at hand to reference, but I won't test you on sort of this kind of a diagram. I don't like diagrams. Okay, so next we're going to talk about the paraventricular nucleus, the PVN. So if we had looked at the ARC was where we started, now we're going over here to the PVN. Okay, and so this is a part of the brain, and it's going to, of course, regulate eating and metabolic processes. Same basic idea, but how does it do that? So this isn't just eating, it's also going to do the things that eating helps. So things like regulating our body temperature. That's related to our sort of use of energy and maintaining that temperature in the body. But it's also going to decide how much fat we store and how much that our individual cells are burning. So it's going to kind of regulate metabolism and fat storage and body temperature and all of that all in one place. So it's a connection, so it has a connection with those ARC neurons, or some of the neurons from the ARC, specifically the POMCs, these ones here. So the ones that make you feel satiated, it has an interaction with those. So those would help reduce our eating behavior. It tells us that we're full, specifically by turning on this PVN's satiety neuron. So it's going to send a signal from that ARC to the PVN to say, we're full, you have enough energy, you can stop. And then we would have eating increase when the other ones, the NPYs and the AGRPs, when those are sort of inhibiting the satiety neurons. So again, they're kind of both sides of the same coin. So we either turn them on or off based on the commands that are coming from the stuff we were looking at on this previous slide. So the ARC is our control center, and it's going to send things to this paraventricular nucleus that say, okay, you're full or you're not full. So we have that possibility going on there. Okay. Then we have our ventromedial hypothalamus, another acronym, so VMH. And this is a brain region that's going to produce satiety and increase metabolism. Okay, so same basic idea. How is it doing this? So this is when we have, again, a connection with those POMC neurons that came from the arc, and it's going to turn those on. So when those come in with a signal, they say, okay, we are full, and so it's going to stop the eating process, but specifically this one cares about turning up our metabolism. So this is that part of switching over to burning our reserves because we no longer have nutrients coming in. So this one is going to be not only sort of you're not hungry anymore, but because you're not hungry anymore, start using the existing reserves because there's nothing left free floating. Ooh, yeah.
So this would kind of like trigger the fasting period?
Yes. Awesome. Okay. And so then we can talk about another peptide hormone, so another chemical that the body produces. And so we have ghrelin. And so ghrelin is an orexigenic peptide hormone. So it's telling us that it's the same basic concept as orexin, very similar. It's going to be synthesized in the stomach and released into the bloodstream when we're fasting. So once we're not eating, it's going to start producing this. So while we're fasting in that fasting phase, we're going to have sort of an increase in ghrelin. And in my intro class, I show them one of those sort of bite-sized descriptions of different processes. And there's a very annoying YouTuber who talks about the fact that ghrelin makes you should think of being hungry because it's that grr sound, like when your stomach is rumbling. And I thought that it was really dumb, except that it's stuck with me for 3 years now. So whenever I see ghrelin, I think of that rumbling of your stomach being hungry. So it's going to start signaling that we need to eat. It's going to stimulate appetite because it starts being produced when we're no longer having food in our stomach. And so the levels will get higher the longer that we've had not food. So that's going to be one of our signals to initiate eating. And so there are other things that we would start thinking about, sort of not just on our body levels, but also we have that cognitive side of things too. So sometimes you start eating not because you are hungry, not because of the different levels of chemicals in your body, but because you've now seen food and it's made you feel hungry because you saw it and now you want it. Or maybe you can smell food. Or if you've ever had an instance where you had a big dinner and you're absolutely stuffed, you're completely full, you could never have another bite, but someone puts a piece of pie in front of you and says, oh, just have one bite. And before you know it, the pie is so good that you'll eat the pie too. So you can kind of push past those bodily signals depending on other factors. And even sometimes when you're not feeling hungry, you might know that it's time to eat because it's been long enough, so you eat anyways. So there's all of those psychological factors as well in the background. But we're focusing more on the chemical side of things here. Okay, and I'm just making sure I didn't want to say anything else about okay, we're good with ghrelin then. All right, and so then this was the diagram again, useful, lots of nice connectors, that is helpful. So that was the starting a meal, that was setting us up to want to eat. What about when we end a meal? What tells us that we're full? So to pick that sort of optimal level of fullness or satiation, it's a pretty complex interaction. Again, hunger is our really, really complex system that we're looking at now. So there's lots of moving parts to this, but it's going to be sort of partially our mouth and our taste buds and sort of what we've been eating. It's also going to be our stomach and how full our stomach is and what's in it, as well as our intestines, which are actively absorbing nutrients and stuff as we're eating. So we're going to have all of these factors that have to interact with one another. And so all of them are going to work together to tell us when we're full. So we know that the liquids that are in our stomach don't get released all at once. As we said, there's sort of a gradual trickle into our duodenum, so a gradual trickle into our small intestines, because they can't handle all of the food all at once, especially where we're wanting this process to sort of take as long as it can so that we get as much as possible out of our food. It makes sense to do it in a gradual process. So that makes sense. So we have specifically stretch receptors around our stomach, or distension receptors. So as our stomach gets full, our body is capable of picking up on how full is it. So if it's really full, it will stretch and expand, and that tells our body that, yeah, you've eaten enough, your stomach is full enough, you should be able to stop. So it's going to send a signal through our vagus nerve up into our medulla. But of course, just having a full stomach isn't enough to completely remove all of our sort of drive for food. It isn't alone sufficient to tell us to stop eating. So we have other things. So the mouth factors that we were talking about can also include the fact that you kind of passively keep track of how much you're eating. So maybe you're used to seeing a certain amount on the plate, you're used to chewing enough or a certain amount of times, or consuming food for a certain length of time. So all of that kind of ties in as well. So there's some of that. We have learning, we have what's actually passing through your mouth. There's something interesting where our body seems to be very quickly aware of the calorie density of what we're eating and drinking. And we'll say that we're full a lot faster if we're eating or drinking something that's high in calories than low in calories. So very, very quickly, if we're having, say, a protein bar, we might not eat a large volume of it, but our body can tell us that we're satiated pretty quickly because it has all of those nutrients that we need in a very dense form. But if you're eating something like potato chips, you could eat the entire bag and it could take a really long time, and the volume might be maybe significantly larger, but because it's a lot lower in terms of nutritional value, your brain wouldn't register that you're full until much later on. So there's all sorts of moving parts that tell us when we are or are not full. So just some things to consider. If we want to talk more about different hormones or peptides, or I guess peptide hormones, so chemicals that are telling us when we are full, the big one here is CCK, or cholecystokinin. There we go. Cholecystokinin. Early morning classes are just the worst for this. But that's our peptide hormone that lets us know that we're full. So that's the satiety signal. And it's going to be released by our stomach and small intestine as food starts moving from our stomach to the small intestine. So it's not only going to help limit our meal size, so it tells us that we're full and tries not to get us to overeat too much, but it's also going to help in the process of digestion. And specifically, it's going to aid in digestion by starting the release of bile and enzymes and all of the stuff that's involved in that digestion process in the small intestines. We don't go too much into that side of things. But so if you had, say, injections of CCK, this would trigger you to eat less during your meals because your body would be told a lot sooner that you were full. And so in a lot of the sort of metabolism type research that's done in rats, they'll try injecting different of these hormones and seeing what happens to the rats. So injecting CCK would lead them to having smaller meals and eating less often because they're going to show up as full a lot more quickly. Okay, so next we can talk about peptide YY3-36. We don't need the whole thing of it. We can just call it PYY. So another peptide hormone, And this is something that's released in our intestines, again, in response to food. So as food moves into our small intestines, we're going to release this hormone, and it's going to suppress appetite. But whereas CCK was sort of an immediate telling you that you're full right now, stop eating, this one is more of a long-term suppression of appetite. So this is, you're still digesting food, so you're still full. We might have moved all of the food out of your stomach and into your intestines, but we're still busy with it. There's still lots of nutrients being processed, so you still don't need to eat anymore. And then we have leptin, and this one is secreted by fat cells. And this is an even longer term signal. So basically the idea is that those adipose cells, the more fat that you're storing, the more adipose cells that you have. Also, I think the larger they get as well. But having more and bigger of these cells means that they're able to secrete more and more hormones. So they would be telling your body that, like, hey, I'm here. And so the more of these fat cells that you have sending that signal, the more your body knows that you have a pretty decent store of existing reserves. And so if you have existing reserves, then you really don't need to be overeating to create reserves because you already have them. So this is a long-term signal, so over the course of, say, days, weeks, and months, that reduces the size of your meals and your daily intake. So if you have high levels of leptin in your body, it's telling you that maybe you could eat less. You don't have to eat as much. Though, of course, we know from experience that just because we have a little bit of extra fat, it doesn't necessarily mean that we start massively adjusting our diets. There's all those extra learned aspects. But if we're just looking at the chemical side of things, then this is telling us long term that you have enough fat stored, you don't need to go out and eat extra. And then, of course, low levels of leptin would say, I don't have any fat stores, you should go out and eat a lot so that you can make some. So this would be really important in finding that set point for our body weight. So finding that good balance where our body is happy and has enough energy stores to function, but isn't too intensely storing food. So somewhere right in the middle. And again, with leptin, if you inject leptin into rats, you should find that they will eventually have smaller meals. They'll eat less overall because that's a signal that's saying that we have enough fat stored away. Okay. And so then this chart is a really nice way of showing sort of some of our stimuli and then the pathways that are involved and sort of condensing all of that information that we've gone over, but into a nice, concise place. So this might be a good spot to start. Say if you're studying, to work out with this, and if you're a visual person, maybe include some diagrams with it. I know that I really liked drawing stuff out to help remember orders. But you don't have to remember locations, that's fine. But these are just sort of your pathways and what's going on when different stimuli are occurring. So a very useful resource. And that's one of my favorite things about this text is that it gives us it does a good job summarizing. And even our chapter summaries do a good job of this too, which is great. Okay. And so that continues. So we have, we have sort of starting eating, stopping eating, and then longer term signals. So the basic chunks we were looking at. So that's pretty good. All right. Let me just do a quick check of how many slides we have left here. Okay. Because the next, we have 6 slides left, but they're all on sort of the same topic. So I'm wondering if we should wait and do those on Tuesday. That's probably for the best. I also feel like I might have thrown a lot of information at you guys today, so that's a lot of new stuff to learn. So maybe we'll end with that nice summary table, and then we'll talk about obesity and different disorders that relate to eating and food intake and all of that on Tuesday, and that'll put us still well ahead of the curve to be ready for our midterm in like a week and a bit. So I will leave you guys there. I hope you have a good rest of your day, and then I'll see you on Tuesday.